Thank you very much, Mabel, and thanks very much to all of you for staying. This is a much bigger crowd than I thought would be able to hang out this long. So as Mabel said, I want to tell you some about the work we've been doing looking at comorbidity of reading and language impairment. Okay, thank you. Is this better? Okay. I read somewhere I was supposed to have a disclaimer, so that's my disclaimer. So we've talked a lot about language impairment and reading disability. Both language impairment and reading disability have pretty good heritabilities, meaning the proportion of the variance that you see in each one of those traits is over half of the variance is due to genetic factors. That also means, of course, that there's environmental factors feeding in as well. But when you have a pretty good heritability, you've got a better chance of finding the genetic causes. So that's why reading disability especially was very helpful in establishing some of the first gene localizations. But there's also comorbidity. 43 to 50% of children with SLI develop reading problems. 55% of children with reading disability also meet criteria for SLI. And 25% of children served by speech-language pathologists are also served for reading disability. And I think these things are important to be aware of. So there are possible genetic overlaps. We've seen some of these genes already in that a few of them have been associated with both reading or language. But often it's like in language impaired kids looking at reading phenotypes, or in kids with reading disability maybe looking at language phenotypes, although there's fewer of those studies. But for KIA 0319, for example, we have found associations for both reading disability and SLI in independent populations. CNTNAP2 or CatNap2 associations have been found for SLI and for reading phenotypes in SLI. There's SLI1, which was the locus identified for language problems, and particularly two genes within that region. Association has been found for reading phenotypes in an SLI population, and PCDH1 is a gene on chromosome 13, and that's one that Chris Bartlett has studied, and he found association with reading phenotypes in SLI families. So some of the problems in trying to look at comorbidity and genetic relationships have been that different diagnostic criteria have been used across studies. That sort of hampers replication within phenotypes or within reading disability and within SLI, as well as between the two disorders. Different genetic markers have been used in different studies, which also gets in the way of replication. Only a small number of candidate genes have been involved in most of these studies. And as Sylvia said, sample sizes have often been too small for genome-wide studies across both of these, although this is getting better. But so far, the genome-wide association studies that have been reported haven't come up with really strong results because of the population size problem. So what we wanted to figure out was, do the same genes influence both reading disability and language impairment, and just how much overlap is there? Is it because there's a common developmental or neurological pathway that serves both of them? And you can take this to extremes. You can say, are all the genes going to be the same? Are these just what we call generalist genes that are at the base of any neurodevelopmental disorder? And then maybe what differentiates whether you're going to have a reading problem or whether you're going to have a language problem might be other environmental effects, for example. Or are they totally separate genetically and developmentally? And I think 10 years ago, we may have thought that these were really two different entities. We were finding genes for reading disability, but not for language impairment as much. And I think that's more, there may be some genetic differences between them, but it may be more overlap than we thought maybe 10 years ago. And I think clinically, too, people were, this is reading disability and this is language impairment, and they're two different things. Are there some genes that are similar with others providing differentiation? I think you've seen some evidence towards that already. And what do the answers to these questions mean for diagnosis and for remediation? Is there some way we can tie it all back to what's important clinically? So I'm going to be talking about two different studies. First one is based at KU, so this is in honor of Mabel and her group. We've worked with Mabel for some time and with Javier Gayan. I've got his address in Spain now. I understand he's moved, but I couldn't find it anywhere on the internet, and I haven't been able to get hold of him to know what. But so recently he has moved, but LinkedIn and everything, he's still got this address. The participants in this study were between 3 and 12 years old, and Mabel has talked a lot about these already, referred by school-based speech-language pathologists. So the proband, the index case for each family, was referred by somebody who had diagnosed them as having language impairment. This was confirmed with regular testing that was appropriate for the child's age. Then we also tested the siblings between the age of 2 and 17. This is whether or not they were affected. So in this, we've got a mix really of a whole range of degree of severity. And I think this gets into some of the things Sylvia was saying, too, about using quantitative traits and getting a little more idea of what's across the whole range of variation. So these siblings, some of them were affected clinically. Some had problems, but maybe not as severe as the proband. Some have had no problems. We got DNA samples for participating children and parents. These used to be blood samples. We've transitioned over to saliva samples because they're much better. If you get involved in studies with buccal swabs, I would recommend against it. They don't tend to hold their DNA as well. So we're primarily doing saliva samples now. And what I'll be talking about today is results from 375 people from 78 families. So this, again, is pretty small. But I think the results are pretty interesting. Oops, went the wrong way. And then these are the phenotypes we used, and we really looked at a collection of phenotypes, partly as an exploratory thing, to look at the heritabilities with each one of them that Javier has been working on, but also to see which ones might fall into certain genes, if there might be differences. These are all pretty highly correlated, but they do get at some differences perhaps that might be genetically mediated. So we have a group of language measures, sort of an omnibus language, and you'll see that popping up sometimes in the results, which is just an overall language ability as on standardized tests. The grammatical impairment measures that Mabel has talked about, vocabulary, mean length of utterance, and then some really interesting measures that Mabel has been working on, looking at trajectory of development of vocabulary. And I think these go very much along with what she presented earlier about how there can be differences in the trajectory of development. And these seemed like these could be very salient markers for genetic studies. We also looked at speech, so this is an articulation measure, the GFTA, and we also looked at reading in these families. So this population, we really have the gamut of phenotypes. Then in another reading study, this is the Colorado Twins study of reading, which has been going on for a very long time, and is now part of the Colorado Learning Disabilities Research Center, which has been funded for over 20 years now And really the reading studies at the University of Colorado at the Institute for Behavior Genetics have been going on even longer than that. So right now we have a set of collaborators that are part of the Colorado Learning Disabilities Center. So we have Boulder, we have University of Denver, and then the lower one is Yale University, which is now part of this as well. So I think all of this fits into the overall theme of teamwork, as you can see. This has really been a multifaceted study, and these are some of the people that are involved in that at IBG, Richard Olson, John DeFries, and Eric Wilcutt. Eric works primarily on ADHD, but also on reading. Richard Olson is one who's developed a lot of the phenotypes that we use. And John DeFries was the former head of IBG. He's now retired, but in name only, he still works a lot. University of Denver, Bruce Pennington and Jan Keenan. And at Yale, Jeff Gruen, who has been working with me on the molecular side. The participants in this study are a little different. Between 7 and 18 years of age, because of course we have to wait till they're reading age to be able to work with them. And these are also clinically referred by schools. Now this is a twin study, so they're at IBG, the Institute for Behavior Genetics, they're studying both sets of twins and they're looking at heritabilities of phenotypes and things like that. For the genetic studies, we're studying the fraternal twins, so it's like a SIB pair study. We can study one identical twin as long as we want them to have a sibling that is also available to study. Because we've been working with pairs. You don't need to for association studies, but it makes it stronger. So we've been doing that. We also recruit other siblings, and again, this gives us a pretty wide range of degree of affectation or degree of severity when we're studying the siblings. So when they look at the overall distribution, especially for the reading disabilities sample, it looks pretty normal because we have so many siblings and everything involved. So this one is a little bit larger. We have 1,266 samples from 308 families that I'll be reporting on. These phenotypes should look familiar. They're very much the same as what Sylvia has used, and this has been very nice that there has been some convergence of phenotypes across reading disability. These are phenotypes that Dr. Olson worked on for years and had been shown to have high heritabilities in their twin studies. So that was really, out of all the reading measures they gave, these are why these are used now, and we've been using those with pretty good success over the years. It starts out with a discriminant score, which is a weighted combination of reading recognition, reading comprehension, and spelling. And so that's been kind of used as a diagnostic criteria, but it's a quantitative trait. And so again, for word recognition, non-word reading, orthographic coding, and phonemic awareness, these are all quantitative traits. We also have what's called endophenotypes. These are things that are maybe not related directly to reading, but might be involved in the process. Perceptual speed and rapid automatized naming. We also have been getting measures on ADHD, and I'm not going to talk as much about ADHD now, but we do have these measures for a total score, inattentive and hyperactive. So then we've been looking at candidate genes for a couple of reasons. We wanted to see how they meshed across across reading disability and across LI. We also have ADHD kids, and we also have some with speech sound disorder. But again, I'm not talking about those today. But so we had selected a set of candidate genes that we thought would cover all of those, and then we wanted to see which ones might be shared. You know, is it all of them? Is it none of them? How would that pan out? Plus, one of the things we're doing in looking at these candidate genes and looking at associations across these genes is this is going to guide our sequence analysis, which is our next step. So we want to try and find if there are specific mutations within these genes and particularly in regulatory regions that might be driving these. And we expect these to be more the rare mutations that were discussed already, rather than the markers that we're using, which are common. So this may help us get down to understanding the genes themselves and how they may be working. The problem is when you sequence these genes, especially regulatory regions, you can find a lot of variations, but they're not necessarily going to mean anything. And by having associations on top of that, we're hoping that that will help us prioritize which ones to look at. So there's a couple of reasons why we chose to do it this way. So anyway, candidate genes that sort of were suggested by language impairment studies were FOXP2, CNTNAP2, PCDH9, and we also looked at CMYP and ATP2C2, which are the SLI1 loci. For RD, some of these are pretty familiar too. On chromosome 2, C2ORF3 has been suggested. ROBO1 has been suggested for reading disability. We also have previous work that showed association with speech sound disorder, and the group at Case Western also reported that. For chromosome 6, this is one that we've been interested in for a long time, and there's a whole collection of genes in there, particularly DCDC2 and KIO319 that you've heard about already. So we were looking all across these genes, and we also looked at DYX1C1, or the dyslexia 1 candidate gene 1. And then we also looked at some that are traditionally associated with ADHD, although a lot of them have association with other disorders as well. But so the dopamine-related genes, serotonin-related genes, adrenergic genes, and some of these were initially investigated years ago because of their relationship to medications that had been given and seemed to be effective. And then some other genes that have been associated with neurological disorders and with ADHD. So those were all ones that we looked at. As I mentioned, we were getting samples, either blood or origin kits for saliva. We extract the DNA. We genotyped 436 single nucleotide polymorphisms across these candidate genes. We particularly looked at regulatory regions and known associations that had been reported before to see if we saw these across these different phenotypes. This is just a picture of the lab and the sequencing machine, or the genotyping machine we used, an Illumina Bead Express. As Sylvia said, these things are developing very quickly. We got this a couple of years ago, and it was a new machine, and we were really happy with it, and it's already considered obsolete by Illumina. And probably because people are going to sequencing so fast, and I'm sure Illumina would like to encourage us to go to sequencing so fast if they don't get outdone by another technology. So it is kind of technology-driven, but as we've worked on this, the SNP genotyping has been very helpful in getting an idea of where we might look in a gene, as I said. We used association analysis. We used a program called PBAT. We Z-transformed phenotypes. We have a null hypothesis of linkage without association rather than no linkage and no association because these are candidate genes. So we thought we should at least acknowledge that. We used an additive model rather than recessive or dominant because we don't really have good evidence now that we would expect a dominant or recessive model. And basically what this association analysis is doing is it's looking at all these SNPs or all these little polymorphisms or variations in the DNA code at specific points in the DNA. We're testing the extent to which siblings with similar phenotypes are inheriting the same genotype. So it's really just a matter of if both siblings have severe reading problems, you'd expect them to be more alike genetically. If they're different, you expect them to be more different genetically if that particular SNP has anything to do with the genesis of the problem. If that particular gene or that particular SNP doesn't have anything to do with it, you'd expect just random inheritance patterns. So we're just testing whether they're different. And this is why, you have to have fairly large numbers to be able to get this to be statistically significant. And as Sylvia mentioned, you have to have correction for multiple testing. So obviously we've got lots of phenotypes, lots of SNPs, a little bit smaller population. And so what we're going to be doing is a gene-based false discovery rate correction. What I'm going to be showing you now is before the false discovery rate has been applied, because that analysis is still in progress. I was hoping it would be done by now, but it's not. So what I'm going to be talking about is the raw data. And I think we can still see the patterns of how the SNPs fall out. The difference is some of these are not going to survive correction. The patterns will still be there, but some of them, we aren't going to know for sure which ones are actually statistically significant. Sometimes things that are just below the level of significance are still important, and I thought Sylvia's description of their gene that they found with laterality on the first pass was below statistical significance, but still looked very interesting, and that's the kind of patterns that we'll be looking at today. So these are the results, basically what we have with SLI, the first couple of genes. Again, I'm not expecting you to care much about which SNP it was or where the position is or which allele. But I think you can see that there's some interesting results across a number of the genes. We have the p-values at the end. Again, these are uncorrected, so they may be, some of them may be a little inflated. We also have what looks like negative p-values, which seems a little strange. This is something that you can opt for in the program, and it's really just telling the direction of the genetic effect for that particular allele. So like in this run, I only did looking at the minor alleles. That means the ones that are less frequent in the population. The idea is that the minor alleles are more likely to be a causal allele, for something that's a rare problem. And so the minus sign might indicate that actually it was the major allele that was associated. And if you think of it genetically, that we're probably not looking at SNPs that are causal. These are SNPs that may be near a mutation that's causal. And so the SNP itself really doesn't matter whether it's a rare variant or a common one. It's just the SNP that happened to be sitting on the DNA when the original mutation happened. And so that SNP gets passed on with the original mutation. And so that's why in a population you find a given SNP associated with a disorder, but it's not causal at all. It's just in there for the ride. And so it doesn't care, when the original mutation happened, it doesn't care if the SNPs around it were common or not. So we have some things associated with the common allele and some associated with the rare allele. What's important is across phenotypes and across disorders is the same allele associated? Are we looking at the same maybe ancestral mutation or not? Are we looking at separate mutations in the same gene? So I thought I'd better explain why some of those are negative. But I think what's interesting about this is looking at the phenotypes that pop up. Even though we did a lot of phenotypes, we're seeing reading phenotypes coming out for what are basically reading candidate genes. And we're seeing a number in DCDC2 and KIAO319 with this PPVT trajectory measure, which I think is very interesting. FOXP2, we're also seeing associations with SLI, CNTNAP2, dopamine beta hydroxylase, DRD2, SLC6A4, which is a serotonin, you may know it as 5-HTT, and then SNAP25. These are all neurologically based things. And again, some with the grammar measure and some with reading. So I think this, the fact that it seems like there's a fairly small subset of the phenotypes that are actually showing association may point us into the types of phenotypes that are important. For reading disability, again, some of the same things, but a little bit different in distribution as far as associations. We didn't get anything with Robo-1 for reading, for example, although we did for SLI. DC-DC2, we actually see more for reading than we did with SLI. KIA 0319, we also see in both of them, but maybe not as many SNPs associated. FOXP2 a little bit, but more with CNT-NAP2. DRD2 again, but not as many of the dopamine genes with RD as we saw in SLI. Some of this, you know, I have to have the caveat that some of this could be some randomness, but what we're trying to do is look for the patterns, and I think this may guide this, at least in hypothesis generation. So overlapping genes that we found for both disorders, the dopamine receptor 5, the dopamine transporter, DC-DC2 is also known as double cortin 2, initially described for reading disability, involved in microtubule formation and neuronal migration, as Sylvia was saying. There's an analogous condition called DCX that is an X-linked disorder, and mutations in that, the serious mutations in the coding region of that cause a genetic disorder of lissencephaly, where the brain basically doesn't have the convolutions that you usually say. It's a smooth brain and with severe disabilities. So clearly a severe neuronal migration problem. KIA 0319 also, as Sonia was talking about, involved in neuronal migration, and ciliopathies apparently could be behind that part. FOXP2 and CNT-NAP2, which has been related to a lot of things. So some of this you've already heard about. I wanted to show, this is, it's sort of a Manhattan plot, but since the markers are fairly sparse and we're just looking at a small part, I've called it an Omaha plot. But so along the base, along the x-axis is basically the location along the DNA, and you can kind of see below the indications of where the genes are along there. Oh. Yeah. There it is. Ooh. Yeah, okay. All right. So this is going along chromosome 6, starting with this NRSN1 gene, which is really right here. This is DC-DC2. KIA 0319 is in here. And them2, or ACOT13, is here. And this is the log of the p-value, just to make it a lot easier to graph. And so it's not a LOD score, it's just the log of the p-value. So this would be equivalent to 0.001. And so you see a little bit of a peak here for SLI. Well, here I can. And a little bit here as well. And so this corresponds to the regulatory region of KIA 0319 that has been often described with SLI and where we have previously seen a signal. The same thing for reading disability, same genes in that we're getting some signal with DC-DC2 and KIA 0319, but slightly different part of the gene that may be stretching things. But it's something, again, as we sequence through there, that will help us judge whether there really is a difference, maybe in this region as opposed to over here where we saw things more for SLI. And again, with KIO319, there's a slight shift over. Maybe that means that slightly different mutations would be involved with reading disability as opposed to SLI. So this is for FOXP2. Here's the gene across here. Now, most of the things like the KE family mutation and mutations for dyspraxia have been found in this region. And you can see some indications here. These are still pretty low. But where we find the most, and rather consistently, has been clear up at the very end of this isoform of FOXP2. And this, we're wondering, could be a regulatory area, too, that we really want to look at. And then as well as I said, kind of in this more typical area that's been associated with more dyspraxia of speech. Reading disability phenotypes, on the other hand, we don't see this big peak over here, and we see that same little dot there, but it really is not standing out. It's up there, but not standing out a whole lot from the crowd. I'd feel more confident in that if we had a few other phenotypes up there as well. So that could be a fluke. The fact that it's in about the same place as the SLI and where other things are make it suspicious. I think if I didn't know that that region was important, I might discount that. CNT-NAP2, we also find a little bit more collection over in here, a peak up here that, again, I'm not so sure that that's going to hold up to scrutiny, but definitely some involvement that, as we would expect for SLI. The thing that surprised me is we got even more with reading and really getting up above a log of 3. So this may be that in our population at least, and it could be that this is a difference in the size of the populations, that we do have more than twice as many families with RD, and so maybe we're getting more power to see some of this. So we have unique genes for SLI as well. As I mentioned, ROBO1, or roundabout 1, which is involved in axonal migration, had been reported initially in a single RD family, but there's been more support for speech sound disorder actually for it, and that may be why we're seeing it more with SLI than with reading disability. Some with NRSN1, it's hard to know if that's a spillover from DC-DC2 because they're so close together. But it could be a gene on its own as well, and has been associated with ADHD as well. DBH, dopamine beta hydroxylase, SLC6A4, as I mentioned, the serotonin, and the SNAP25, where we got more in this than we did with RD. The only thing that popped up with RD that we didn't see with SLI was ATP2C2. And again, that was only just a few SNPs, so I'm not sure if that's real or not, and that'll take more follow-up to find out. So this is just an example to show what it looks like when we really don't have much of a replication at all. This is for the serotonin receptor. We get a pretty nice signal. It looks, that looks real to me because things are kind of stacking up there. And RD, you know, maybe you get one blip here, but just nothing the same at all. So I think this is more of a discriminator. One of the things we wanted to look at then across these genes was how do they relate to each other? Can we use this as in kind of a heuristic way to look at new pathways? One way to find novel genes is through a genome-wide association. Another way is to try to define pathways. And this ingenuity pathway analysis is a program that's been developed to try to use what's known in the literature to make pathways between genes and to kind of show you where networks might be. So it might show us other genes then that we should investigate to be able to see if we can define some pathways. So we tried this with the genes that we got for reading disability and that we got for SLI to see if they would overlap at all. Since many of the genes were in common, we thought maybe we would see some overlapping in the pathways, but then again, maybe we'll see some differences. And I think with the SLI, since we had a lot of genes that just had 1 or 2 SNPs, I only included in this analysis ones that had more than 1 SNP involved. So I cut down the number of genes a little bit to the ones that would seem like they might be the most likely to be associated. And that's how, this was the pathway we got out of it. Again, these are going to be complex. But what I thought was really interesting is here's dopamine here, and we have a couple of genes, including FOXP2, feeding into that, and of course, the known dopamine genes, DBH is in there. But I thought that was, it was neat that it came out as a node for our population. Some of the others you can see here. This is a fairly large node, but this is ubiquitin, and that's kind of a general pathway that just about everything feeds into, so I'm not as sure that that means that these genes are involved directly with ubiquitin. It's just a protein salvage pathway. But it does show some connection. The one thing that was sort of disappointing in this ingenuity analysis is that it doesn't seem to want to draw the connections between FOXP2 and CNTNAP2. That was a little, so it's not a perfect system, but it did confirm this association with FOXP2 and dopamine. So it's amazing that this correlated so well with the previous talk. And actually with reading disability, we got a little bit different pattern. Again, nothing so much with dopamine because those genes weren't as significant, although we have some of them up here. But the kind of nodes here, HTT is Huntington, which is, although it's involved if you have the right kind of mutation with Huntington disease, which would be quite a different thing, it actually is involved in very early neuronal differentiation in setting fate, whether you're going to proliferate or whether you're going to develop into a neuron. So it really is kind of interesting that this kind of popped up, and maybe that's something involved in the mechanism. And this gene down here with DCDC2 feeding into it and CNTNAP2 is also similarly involved in early gene differentiation, neuronal differentiation. But again, I wish they would have drawn a little bit better connection between FOXP2 and CNTNAP2. But I think this might give us some conceptual things to look at in the future as we try and put these pathways together. And I think the work that's being done on individual genes in generating their upstream and downstream interactions, it's going to make pathways like this a lot more robust, where you'll see things that aren't just represented by what's in the literature, but actually have a lot more value behind them. So I think these programs, while they're in their infancy now, are being developed more and more, and will help us in trying to figure out what the basic problems are and how these genes might all interact together. So despite having a number of the genes in common between these two disorders, we did see some difference in the networks. Whether or not this is reflective of alternate pathways, we'll have to see and verify it with our gene-based analysis as well as we go through and try and look at a gene-by-gene correction. So our conclusions are basically that SLI and reading disability do show overlap in some of their candidate genes, but other ones seem to show differential influence. SLI tended to have more influence by FOXP2 and dopaminergic genes. Reading disability in this was influenced more by CNTNAP2, which I'm still surprised at, and may be more influenced by early developmental genes. And as I mentioned, we'll be using these results and the locations of the influence to guide our DNA sequencing to actually try to find causal mutations. Then we'll be taking that again into other populations to see if those same mutations are involved in other groups. Our populations have been pretty much Caucasian, and Jeff Gruen has been collecting families of Hispanic and African American background, and we'll be testing to see if some of the same variants that we find in the genes that we're studying in the Caucasians, whether those translate over, whether it's the same variants or whether we'll find the same genes with different variances in different populations. So what are implications for practice to try and get it down to what we're all after? I think part of this is in diagnosis. We'll need to consider that many children show elements of both SLI and reading disability, and that this may be a primary disorder, that it's not just that coincidence that some kids have reading disability and have SLI as, the overlap of two common disorders, or that having reading disability makes you more likely to have SLI, or more likely the other way, SLI makes you more prone to reading disability. But it may be that there are actually some common factors that affect both of them. And maybe this overlap is because of primary causal factors that they have in common. It certainly would mean, and I think a lot of places are doing this, but including language testing for children with reading disability and reading testing for children with SLI. But as we develop this more, it may become clearer that this is really more of a continuum rather than distinct disorders. And I think that can affect some of how things are practiced. In the past, at least, when I was working on multidisciplinary teams, there was the speech-language pathologist and there was the LD specialist. And they went at things completely differently. And maybe we need to see things converge more. Another question then is, will we ever be able to have gene profiles assist in the diagnosis? And this is a question we get a lot. My reaction to that is actually very close again to what Sylvia said. Somebody comes at you saying they've got a test for reading disability, don't believe it. And I think it may be that within families we will find genes, and we may find mutations within certain families that would help you be predictive. But with all of these, there's environmental influence as well. There's interacting genes that we're not looking at. So I would actually be very cautious about thinking that we're going to be able to do genotyping and tell you whether your child's going to have a reading disability. We might be able to say they're a little more susceptible, but in some ways, knowing the family history and knowing some of the early phenotypic expressions, how's their language doing, might be more predictive than actually what the genes are, at least at this point. maybe when we're able to sequence everybody's DNA and everything, things may be different. But I think at this point, I don't think that's where we're headed, and that's not really why we do these studies. We're not looking for the gene for or the mutation for. It's more seeing these pathways and seeing development, and then what can you do when you understand the underlying development. So are there implications for treatment? Again, children with SLI and RD could benefit from speech-language approaches as well as LD approaches, and a need for the coordination of treatment. And it may be that there's still things like DSM-5 now that are going to complicate this. You know, if we're going to see some of these as a continuum of communication disorders. Another question then comes up, could targeted medication be a useful adjunct to therapy as they do with ADHD? Well, we're finding things with dopamine, there's medications that might help. You know, maybe there will be some things. And as was also mentioned earlier, you know, if medications help something that a gene that's involved, like CNTNAP2, it's involved in schizophrenia and it helps schizophrenia, does that mean it might also help with language problems or something? I think that remains to be seen, but it may be that that type of approach, as we understand the development, we understand what the genes are doing, maybe we will find targeted ways of helping as an adjunct to therapy, helping kids be able to learn a little bit better. I don't know whether it's going to be by changing their acetylation or what, but it could be that there will be processes that we'll understand better once we understand the underlying development. So these are my acknowledgments. Again, it does take a team to do all this stuff. So the University of Kansas, the University of Colorado, the University of Denver, and then our lab at the University of Nebraska Medical Center. And these are the people in my team. James does a lot of the genotyping. Judy does manages the lab and does a lot of the setting up of DNA processing, and Denise Hoover is in charge of keeping all of our data organized in a rather large database. Plus, everybody does everything. So, that's it. Thank you very much.
Some questions? Yes.
So we work in Fragile X syndrome, and from reading your article that was sent out to us, we were wondering if the similar phenotypic language impairments that are seen in Fragile X that are similar to autism and specific language impairment, especially in boys, can those genetic markers or information from Fragile X be used to inform the identification of the genetic basis of ASD and SLI?
Yeah, Fragile X is very interesting, and actually I haven't been working with that, but some people in my department are, and the mechanism for that, at least what Dr. Dunevsky has been working on, has to do with dendritic spine development and everything, so I think that's a process that we haven't studied as much in language impairment. I think that could be very interesting. I know that the types of language disorders between Fragile X and SLI, my understanding is that they're a little bit different, and Dr. Rice is much better on that part of it. But whether, certainly synapses and everything, I think, will be involved. And so I think there's some overlap there. And it would be interesting to know if we see some of the same dendritic type manifestations in genes involved in language impairment. I think that would be very interesting. And some of the genes, again, that my colleagues are working on may help answer that. I'm not aware of directly genetic evidence.
Hi. I was wondering, are you aware of how the prevalence of reading disabilities and language impairments compare to other countries? I say that because as a nation, I think we are exposed to a lot more mutagens compared to other countries where GMOs and things like that are banned. So do you have any insight on how prevalent those kinds of environmental factors, how they affect, neural development in those countries compared to here?
The types of things that we're looking at probably wouldn't be related. We wouldn't be seeing them as something that is not acting directly on inherited DNA. So I really couldn't answer anything about level of mutagenesis. What seems to be more of a factor between countries is their linguistic system. But it is seen all over. It's maybe more pronounced in English because people have such a hard time learning English. It's orthographically a mess. But when you look at orthographies that are much more transparent, not only do you still find dyslexia in those populations, but they also find some of the same genes. Like Italian, as I understand it, is very nice as far as spelling and everything, and yet they find association with the same genes that we see here. Finnish is another one that is orthographically transparent. And they also see some of the same things. The ones where you may see a little bit of difference are in China because their orthography is not phonologic. And a lot of what we study, these are phonological phenotypes. And so that sort of biases us towards genes that involve phonology. And so we may see different things, different patterns of brain activation and everything in Chinese, but then when you go to Japan and they also have a phonologic method of orthography, that changes as well. But I think we're seeing things pretty much when you control for the orthography, the way the language is written, it's pretty consistent.
Dr. Smith, you mentioned possible medical therapy.
Oh, thank you.
Medications or drugs. How far in the future would you imagine that might be?
Well, I don't know. I was looking at a paper. I think it'll be a matter of testing them out. I don't know that it would be all that far in the future because there's so much work that's being done in other disorders, on small molecule therapy or on neurotransmitters and everything. I saw a paper where they were talking about a disorder involved in autism that was due to a gene mutation in one of the genes on that network. And they found that in a knockout mouse that was missing that gene and had a lot of behavioral phenotypes, that if they blocked serotonin, they augmented serotonin, and that corrected for some of the problems. Again, it's a pretty gross analysis because we're talking about knockdown of a gene completely in a mouse model and everything, but that you can see some efficacy. So if you just had something where you're decreasing the expression of a gene rather than totally knocking it out, you're giving a serotonin agonist, would that help in some of these behaviors. And that might be why some medications have been helpful in disorders, but some of the medications that have been used are pretty blunt force things too. As we get to understanding things more, maybe there will be more direct targets. Certainly the cancer investigations, I think, are going this way and trying to find what genes are involved in different kinds of cancers and what molecules can directly target that. So I think in some of the same ways we'll be looking at what might directly target some of the developmental problems as well. There's a lot of parallels.
That was very interesting. I just wondered, I know you said that, the gene profiling system, we're not at that point where you can do a gene profile and predict whether someone is going to have a reading disorder or specific language impairment. But do you envisage, I know with, certainly in Australia, there's a lot of work going on in the field of cancer, I think, in terms of, doing genetic testing and being in, do I have the breast cancer gene? Do I have the, there's quite a lot of genes being identified, and so people are getting tested. And so it seems to me it's on the path ahead of us at some point that the same thing's going to happen for speech and language impairments. And I'm just wondering if you've got a sense of how far away that would be for our field. It's a similar question, I suppose, to the medical one, but it's a reality at home. People are certainly getting tested and screened all the time. So it's sort of like an indication of whether you're at risk or not.
Yeah, and I think what makes a difference with some of those is where you've got a major gene that has a strong effect. And even with the breast cancer genes, it's a probability, but the probabilities are pretty high. So if you have a BRCA1 mutation, and you're looking at a 70% probability as opposed to a 10%, that's actionable. That's something that you can really do something about. And other genetic disorders where you've got a major gene effect, that's where you really can do testing. Some of these other conditions where we haven't found, most of these genes have been sequenced in kids with reading disability, for example, and we've never found a coding mutation. So that's why we're looking at these regulatory mutations that are maybe going to bump the expression up or down a little bit. Those are going to be harder to be able to say, well, this mutation gives you this much risk. Maybe if you have 10 genes and they're all down-regulated or something, you might be able to give a probability. But that's where I'm saying it's a little bit harder to be able to give anything more than saying, there's just some increased probability. An example is diabetes. People have been trying to look at biomarkers or look at SNPs that would give you a better prediction of whether you're going to be diabetic. And when they add all these together, the ones that are known so far at least, they don't give you much better prediction than for type 2 diabetes than are you obese? Or what's your blood sugar? So they're really not that predictive. So it depends on the disorder how much genomics is going to help you diagnostically. So I'm just sort of being a little bit skeptical that it may be a while before we get enough information to know which mutations are out there, how much they downregulate a gene, and how many of them it takes to get a prediction that you could really put something on, rather than saying, well, everybody, if the population rate of reading disability is 10% and these SNPs give you a 20% risk, that's an increase. But are you going to act on that, or is early testing maybe going to give you even more information? And in combination with that 20% risk, might push you over the edge to say, okay, we need to do something. But the genomics alone may not be enough.
I can talk really loud. Shelley, would you say a little something about your thoughts, and I don't know whether Mabel might let it chime in, on how much effect you think intervention intervention can have, especially early in life, on the child who has is at risk for reading disability in particular?
Yeah. No, I think it really is important, and it's going to be like a lot of developmental disorders, where the earlier you intervene, the better. The paradigm I always think of is hearing loss, because that's another strong area, and if you get in there right away, you can make a big difference in language development. It may be very similar in this, in that if we get an idea of what the very early manifestations are, and that's why I think studies like Mabel's, where she's following these kids from, from the time of birth, to see what some of the early manifestations are. And there have been other studies to try to see if there's other ways of detecting reading disability very early. There was a study, is that Finnish as well? One of the Scandinavian countries, where they were doing evoked responses in infants and then following them. And these were high-risk infants in that they had a sibling with reading disability. And they were finding later, as those kids grew up, that there was an evoked response signature that seemed to help predict which children at risk were going to develop reading disability, went on to develop reading disabilities. They followed them longitudinally. So there may be things like that. But then again, this was with kids at high risk, just by virtue of their family history. But getting back to your question about whether once you've identified these kids, will early intervention help, and what types of early intervention? Is it working with language and everything in a child who's at risk not for language impairment but for dyslexia? Would that help? I think it'd be really worth a shot, especially if there's that much overlap as we're wondering about now. So I don't know. Do you have anything to add?
We need to learn a lot more about what's going on in those early stages. It's very hard to get this work funding because it's kind of just give us a lot of money to follow all the kids. And today's world, that's probably not compelling, but that's really what's needed. And I think we're going to have to rely on countries and cultures that have made a priority to do these things. And they are in the Scandinavian countries and in Australia that are making a social commitment to paying for the collection of this kind of information. But as soon as we identify children, I believe that the resources are well spent to stay with them. It doesn't mean that you're providing intensive therapy, but it does mean that at the very least you're in a very close working relationship with their families and can offer some guidelines on what to watch for, and when those milestones aren't met, to get back in and do more direct assessment, and also to work with the families on how to interpret these differences. Families get a lot of their self-identity and their emotional regard wrapped up in whether or not their child is beginning to talk, and if the words are coming in on time, and then they start worrying about all of the explanations for why this might not be happening.
I think this does bring up another thing, though, that when we talk about genetics and their influence on these things, a lot of people will, especially lay people will say, oh, well, it's genetic. That means you can't do anything about it. And this is a risk factor like everything else, and certainly therapy is very effective. And so I'm glad you brought that up, because families will ask you about that. And no, the geneticists aren't thinking that we're going to tell you whether therapy is going to work. We're hoping you'll tell us what therapies are appropriate for the types of disorders that we're defining.
My question sort of follows on from that. I work in psychiatry now where we look at gene-environment interactions a lot. So you look at what happened early on and see how they interact with the genetic code. Do you think it's possible that there might be some of these genes that might be more responsive to early intervention than other genes? Is that a possibility, or do you think that they're entirely separate, the language environment, the intervention, and the disorder?
I think that's a really interesting question, and certainly there are people that are studying the kids that are resistant to treatment And that's part of there's a group of centers that are these learning disability centers, and a couple of them are really focused on that. And there is a group of kids that, despite what we think is the best therapy and everything and really intensive, that are resistant, just have a whole lot more trouble. And it may be that we'll find some of these genes more common in that population. Certain genes may be more represented in that population than in the general population. I think that's quite possible. I haven't seen anybody look particularly at that population yet, but I know it's in the works. I think a group in Texas wants to look at that.
One more question I see in the back.
Thank you, that was a wonderful talk. My question is probably a little bit off-centre, but I work also with some degenerative diseases like primary progressive aphasia, and I've heard some discussion about whether or not early language difficulties or reading difficulties predispose people later to a degeneration of language. And so I know that there's opposing opinions about this, but I don't know if any of that has ever touched in your lab, or you've had any thoughts about that area or not.
I can't say that I know anything about it directly, certainly from our studies, but I've wondered about things like that in that where I work, there's a lot of people working on degenerative disorders, and whether those are involved in whether there were early manifestations. I think that could be a possibility. Those families wouldn't be getting into our studies, so that's why I don't know anything about it, but I think it's possible. It's sort of like, again, I draw analogies with reading disability, or with hearing loss, in that some of the progressive hearing losses may also have some other problems that go along with it that may be, if you've got a neurologic function that's degenerating with a progressive hearing loss, is it also affecting other things as well? So I could see that actually being a possibility, that some of the same genes may be involved but differently. Some of the more severe degenerative disorders could be a more major gene effect, maybe involved, maybe other genes, or the same gene with different types of dysregulation might be involved in something more benign, where it's not really a degeneration, but just poorer function. So I think anything that's involved in neuronal development and maintenance could be interesting.
Any other questions? If not, thank you so much, Dr. Smith.